by far not the least, from John Chodera. John is a member of the Computational and Systems Biology program at Sloan Kettering Cancer Center in New York City. His lab is developing the infrastructure to enable fully autonomous small molecule drug discovery that is powered by the next generation hybrid physical and machine learning models. And these models are used to predict activity resistance in a data efficient manner. Something I truly admire and want to highlight is that John has been one of the biggest proponents and contributors to open science, developing, for instance, OpenMM program in Postfield, which has been downloaded for 1.2 million times. He also has been extremely active in creating open science communities. To list a few: Open Force Field Initiative, the COVID Moonshot that made international headlines, I think, Folding at Home Consortium, and more recently, AI-driven structure-enabled antiviral programs. John is a recipient of Luke Gerstner Young Investigator Award and several other awards, and we'll look forward to the talk.
Thank you so much, Alex, and it's a pleasure to be here speaking today. Thanks so much for the invitation. I'm hoping to give you some very practical advice on how to use physical modeling and drug discovery that might be a good foundation for your explorations in this workshop as you go off and do more cool stuff within AI and ML and blending it in different ways with the kind of physical models we're talking about right here. So, just to start, I would like to highlight, the challenge that we're facing in drug discovery is that it's really slow, it's very costly, and there's an extremely high failure rate. I've tried to synthesize all the statistics here, and you can find a copy of my slides, by the way, at our website, choderalab.org, so you can follow along. I've tried to synthesize various statistics about the actual success rates and the costs, not including failures at each one of these stages. And what we're going to talk about today is how we use physical models in these design, make, test, analyze cycles within the discovery part. But of course, the failure rates and the costs are very high for each of the subsequent stages as well. So anything we can do in discovery to have more shots on goal or to have higher success rates is certainly going to help us improve the overall state of drug discovery. So I speak from a little bit of experience. Of course, I collaborate a lot with folks in drug discovery companies and industry who are counterparts there that actually have to do this all day, every day. And I have tremendous respect for them because I've had to sit through a drug discovery program, being the computational chemist supporting this program as part of this open science example called the COVID Moonshot, where it's unique among some discovery projects because we've been able to put all of the data online. So you can actually go to this website and find and download all of the data in a drug discovery program if you really want to look at the nitty-gritty details of what happens in these programs. I'd encourage you to do that, but just as an example, we started from an X-ray fragment screen in February of 2020, where we just had some very weak millimolar, perhaps, compounds binding in this X-ray crystal structure, and through a series of rapid hit to lead, assisted by computational methods, and lead optimization assisted by more normal human medicinal chemistry teams, as well as computation, we were able to progress this towards a preclinical candidate in just about 18 months that had very different liabilities than the other approved drug like nirmatrelvir or Paxlovid that you might be familiar with and maybe even have taken, and Shionogi's improved ensitrelvir as well. So I'll use this as an example as I'm talking about some of the challenges that we're facing. So obviously, in any drug discovery program, you have to have a design objective. That is, in this case, for small molecule discovery, called the Target Candidate Profile, or TCP, that specifies exactly what we're trying to achieve. First of all, for example, you might want to hit a particular target. In this case, it was the SARS-CoV-2 main viral protease, and you have to hit a better affinity than 10 nanomolar in this case so that you can have a pill that is small enough to actually take. We also want it to get into cells and do its activity within inside of cells. That's really important. So there's other properties that are important there. If you want to orally administer it rather than other routes, it needs to be highly soluble and also needs to have a half-life after it's orally absorbed that's long enough that you can have 3 pills a day or fewer, because otherwise it becomes very difficult to manage. And then, of course, there's this huge panel of toxicity concerns that we've heard about some of this already, where we have to make a therapeutic that's safe enough to actually take for 5 or 7 days. Now, the problem here is that we can't simply rely on generative models, even though there's a lot of excitement about how these models can be useful in drug discovery, simply because we don't have the scale of data. Barbara already spoke about, the total amount of data collected in Chembl is enormous, 221 million measurements compared to the number of structures, for example, in the protein data bank, which is only about 220,000, but that's still 6 orders of magnitude away from the number of tokens used to train something like GPT-4, where we're actually seeing these generative models producing useful text that can be useful in certain subdomains. And to get there, to make 6 orders of magnitude more data in drug discovery using traditional methods would cost something around $6 trillion. And it's very unlikely we're going to see anyone willing to outlay that much money to actually get us to that scale. So we have to be much more efficient and much more clever about how we approach drug discovery, especially for target-based properties. So in order to do this, we typically organize ourselves into what's called an assay cascade, where we have all of the cheap and easy assays that come right after synthesis in this first tier. Of course, we still have to make the compound, which is usually very difficult. It costs money, it costs time, and then we have to do some logistics and get it through these assays. If it does well, then we put it through more expensive assays that have limited throughput, and then finally towards more expensive assays that tell us about the pharmacokinetics and consume animal lives, which are absolutely critical for advancing drug discovery, but is something we want to do only when we have to, obviously. So you can find more examples of these assay cascades from our Open Science Antiviral Drug Discovery Program, one of these Avid centers, asapdiscovery.org. You can actually see all of the target candidate profiles and assay cascades for all of our active programs if you want to explore more of what these programs actually look like for a diverse set of programs. But a lot of the time, you're spending your time through these design, make, test, analyze cycles, trying to make some new design ideas, prioritizing which ones you think are going to be successful, queuing them up for synthesis, and then assaying those compounds, hoping you get through each of these checkpoints to get all the way down towards things you might be really willing to put into a very expensive preclinical program to convince a regulatory agency like the FDA that you have something that's safe enough and efficacious enough to put into humans. Now, there's a lot of opportunities for modeling to impact all of these stages. Essentially, what we'd love to have is surrogate models that are highly predictive for each one of these assays. And for many of these cases, we're just looking at ligand-dependent properties like solubilities or lipophilicities or log-D. These are things where we might accumulate enough data to make good machine learning models that are based on ligands across many different drug discovery programs. But if we're dealing with a new target, we might not be starting off with any data at all. And so we might have to rely on, or might want to rely on, something like a physical model that is more generalizable than something that requires ligand-based data for the target, which we may not have. I'm particularly going to be talking about these free energy calculations, which are sort of the industry standard way for using physical models to advance drug discovery by making choices about which molecules to make. And what's cool about these methods is that they are, of course, enabled by using structural information, as Stephen Burley had said earlier today, that tells us about what the binding site looks like and possibly how other ligands might interact with that binding site. And what's remarkable is that in just the last decade alone, the PDB has more than doubled in size just from depositions of structures in pursuit of other science goals. We've seen over 100,000 new structures in just the last few years, which is absolutely remarkable. It's still quite small, 220,000 structures or so, and there's important gaps that remain in a lot of different things like ADMET-related proteins to understand what they look like, for example. And so, but given things like AlphaFold, we've been able to very densely model a lot of interesting targets of interest. So this is becoming more and more of a useful resource where we can use these structural and physics-based modeling techniques to actually answer questions. So what these design, make, test, analyze cycles have a lot of is often trying to either improve potency. For example, this is a very weak lead molecule that we started with, or maybe a hit in the COVID moonshot, 25 micromolar, and trying to improve the potency, or later on when you're working out other issues like metabolism, trying to maintain potency while you're trying to work out changes to the rest of the molecule to change those properties. So you might start with this. You would identify a retrosynthetic route where the chemists have to optimize this route, which often takes some time, sometimes weeks, and may not be possible to get very high yield. So it might limit, or it might cost a lot for each one of the compounds you want to synthesize. But then what you want to do is use the different starting materials in this last step, like Connor was suggesting, where you can then very rapidly have a chemist, single chemist, for example, might make a dozen analogs per week. Now, traditionally, medicinal chemists will choose things that are very conservatively selected, where they're just making very small changes, as you'll see right here, to one part of the molecule to feel out the structure-activity relationships. But in this case, you might be able to have many, many compounds, as Connor had suggested previously, that are compatible with this last step in this reaction, in this case, of palladium catalyzed coupling. In this case, there were 15,000 potential groups that could be installed at this R3 site. You can model all of these and what these might look like in the binding site in a static structure. But what you'd really like to do is to predict the affinities of this compound in a very accurate way so that you can design or select exactly which compounds are likely to be the most successful at improving or maintaining potency. And like I said, it's very difficult to do this with a novel target where you may not have a lot of data for this particular target. So one of the best physical modeling approaches that we've been able to use is called alchemical free energy calculations. I'll tell you more about what the alchemical part means, but essentially what you're trying to do is you're asking, what is the difference in binding affinity between a ligand A and a ligand B, where I've made some small perturbation to that ligand structure? And there's many ways you can run these calculations, but fundamentally they involve this thermodynamic cycle, which means instead of running a simulation and waiting for the ligand to dissociate many times, which would take longer than the age of the universe, perhaps in real time, I can do some very clever stat mech and use this the fact that the free energy around the thermodynamic cycle has to end up being zero, adding up to zero, and instead do two easy to compute calculations, a transformation in the binding site and a transformation in solvent, to compute the overall change in free energy for this ligand. And what you can do is take a look at our best practices for alchemical free energy calculations, which is part of the Living Journal of Computational Molecular Sciences. This is a continually updated best practices review that captures a snapshot of what the field believes is important in running these calculations accurately and robustly, if you want to learn more about exactly how we do this. The cool thing is that this same kind of operation where we're computing a free energy difference in different environments and subtracting them to get the results we're interested in, the property we're interested in, is actually useful for much more than just driving affinity or potency. Not only can we do that, and I've given you some examples of each of these categories, but we can also derive selectivity. For example, if you're looking at two very related active sites, like in kinases that might share a lot of homology, or if you're looking at predicting the impact of a mutation, for example, on your target, which is often the case in cancer, where we will see patient individualized mutations that may change drug response or even sensitize the target to particular drugs. And other folks have also used this for biologics, where they're trying to optimize the thermostability of antibodies, for example, by making mutations that increase the melting temperature of the particular target. There's also other things that are less target-centric and maybe anti-target based. We're getting more structures of things like hERG, this ion channel you heard about earlier, where you'd like to avoid binding to it to avoid causing cardiotoxicity. As we're getting more structures of these, we can use these free energy calculations to also address questions of anti-target binding and compute other ADMET properties. And then also other physical properties like solubility and crystal polymorphs, which turn out to be really important problems. Again, the same technology can be used to address a lot of these questions. The way these calculations work is that you run molecular dynamics simulations where you're actually perturbing one ligand into another by modifying its Hamiltonian in some complex way. And by running a lot of simulations at these alchemical intermediates, you're able to compute the entire free energy for this transformation, including rigorously entropy and enthalpy. So you get a really good estimate of what the free energy difference between these two molecules binding are. There's other variants of this that can allow you to compute absolute free energies, which are much more expensive, but fundamentally they all use some way of sampling all of these alchemical states in some way. There's different methodologies that people have developed. This is just a snapshot of some of them. Replica exchange is currently viewed as the best gold standard right now, but it's a little bit tricky to implement. There's other variants of this, like non-equilibrium switching, which use some really cool recent statistical mechanics discoveries from folks like Chris Drzynski and Gavin Crooks that allow us to highly parallelize these calculations over many GPUs, which is great for cloud computing. And there's lots of folks that are effectively using that right now. How accurate do these calculations have to be in order to be impactful? Well, it turns out they don't have to be tremendously accurate. If we're looking at how Abbott chemists, for example, were making modifications at the hit to lead and lead optimization in pursuit of potency, they're pretty much modeled by a Gaussian here in terms of binding free energy changes. And if you look at the shaded region here, it's the probability that they've improved the potency by a factor of 10. Well, a method that's even 2 kcal per mole inaccurate would increase or decrease the number of compounds you need to synthesize to hit that goal by a factor of 3. And if we can get all the way to half a kcal per mole, you're basically speeding things up by an order of magnitude or reducing the number of design cycles you need by an order of magnitude, which is hugely impactful. So there's a lot of efforts that go through and use tooling in this area to compute retrospective benchmarks where you have an established benchmark set that you've curated, and you can assess the accuracy of these methods on different target classes and different scaffolds. It's highly variable. Sometimes it works very well. Sometimes it works not as well. But overall, the accuracy is quite good. 1.4 kcals per mole is exceptionally useful in terms of prioritizing which compounds you might actually make and getting rid of a lot of bad designs. You can see more about how we construct these test sets to actually address the questions we're answering or asking about binding affinity in this other live comms publication. And we've personally used this as part of the COVID moonshot prospectively to great success. There were a lot of opportunities to make improvements in various scaffolds and a number of important transformations that were prioritized by these alchemical free energy calculations. But this is really working closely with a cast of medicinal chemists who are driving a lot of the ideation and decision-making at each one of these stages. And it's really, of course, takes a team to make all of this happen. You can find all of the data and read all about the story here. It's all open science, like I said. Sometimes, however, things did not go so well. We frequently found that we had mismodeled the active site in different ways and found some of the compounds agree very well, and then there's a huge number of things that just have no correlation between experiment and calculation. This is a more general phenomena. In fact, Christina Schindler at Merck KGaA has written a beautiful paper here that I'd encourage you to check out about the kinds of successes and failures she found in trying to apply free energy calculations to various programs internally at Merck. And has categorized both the kinds of errors she got and the classes of failures that she observed and was able to pin down. In total, about half of the programs worked out quite well. Half the programs were failures for various different reasons. One of the other important things is that the current level of accuracy is certainly an opportunity to improve upon. We can't generally run hundreds or thousands of drug discovery programs over and over again and see what the impact of using a tool is on that cumulative distribution function, but we can do this synthetically in this cool tool called DrugGym, built by Michael Retchen in my lab. It allows us to synthesize or synthetically model what happens if we use tools of different accuracy and see how that shifts the cost in terms of how many molecules we need to make in a model hit to lead phase of a realistic program. And you can see that amount of money that it saves you in terms of getting there faster and cheaper can be enormous as we reduce the modeling error. So there's substantial interest in making these models even more accurate by improving upon their performance. Now, there's three critical areas in which this improvement needs to happen. One of them is the accuracy that the force field or molecular mechanics simulation model is for modeling the interactions between all of the atoms in the system. We've been using these force field forms that are about 45 years old now. So there's ample opportunity to move to more faster hardware. People are exploring neural network potentials, for example, as a way to improve upon this. Also, we neglect a lot of essential chemical effects like protonation and tautomeric states that can actually be quite important in particular kinds of interactions. And finally, there's been a lot of interest in modeling different conformations of proteins that are relevant, especially protein ligand poses that might be relevant as well. And the problem here is that often there's millisecond scale motions, but our simulations are much shorter at nanoseconds to microseconds. All of these are ripe for AI and ML to accelerate our progress in all of these areas to make things better. But to start with, you might want to start with a good open foundation that you can use both as a baseline and a foundation for integrating innovative technologies and assessing them in different ways. And the Open Molecular Software Foundation is a nonprofit that is building this whole ecosystem of open source tools for accelerating drug discovery. The work I've talked about already has been from the Open Force Field, for example, which is building molecular mechanics force fields according to best practices, as well as Open Free Energy. But there's also OpenFold, which is producing AlphaFold-like models in Rosetta Commons, and a new Open ADMET project aiming to predict ADMET properties using new data funded by an ARPA-H grant. Open Force Field has been producing both open source tools and open science data sets, as well as open iterations of force fields that conform to best practices. And you can find out more about their work at openforcefield.org. But I especially want to highlight the work of the Open Free Energy Consortium, because you can use these tools in your own discovery programs and experiment with how you can do better, for example, using other ML methods just yourselves if you'd like to. There's a whole staff of professional developers that are contributing to this, a technical advisory committee consisting of academic members, and a consortium of industry that provides both governance and guidance, as well as funding to make this effort sustainable. They've been working on building code in lockstep with encoding best practices captured from a broad industry cross-section in these Living Journal of Computational Molecular Sciences papers. And what's cool is that you can actually try this in a Google Colab notebook from your browser if you go to openfree.energy and get started playing with these sorts of methods today. Now, there's a lot of tools as part of this infrastructure. There's tools for planning campaigns of transformation networks where you might have a lot of related designs that you might want to score. There's tools for iterating over different kinds of optimal transformations between these ligands because you have to line them in the binding site and map atoms from one set to another. There's also open source tools like OpenMM, as Alex had mentioned, that allow you to execute these calculations very rapidly on a GPU. They currently take about 12 hours per transformation, so they're quite expensive compared to ML-based approaches. There's a limited number of different algorithms that are encoded right now, but it's encoded in a modular way that allows this to be rapidly extended. And the team is working to build more kinds of tooling that you can actually use and improve upon. You can easily swap out a module and see if you can do better, as well as using best practices and encoding those for analyzing the calculations and representing how you assess these calculations in a best practices manner to make sure you're not being deceived by the statistics. There's also tools that are available now called Alchemiscale for orchestrating very large campaigns of these on distributed or cloud computing resources. And we've managed to use these, for example, between the Antiviral Discovery Center, ASAP, and these two projects run over at least 500,000 GPU hours just in the last year alone. So these are getting to be quite robust and quite scalable. The amazing thing is that they're able to do continuous benchmarking using HPC resources to make sure that they're always improving as they add new features or change the ecosystem and make sure they haven't broken something. So there's a lot of effort devoted towards ensuring that they're delivering the best practices to you as part of what's going on in these tools. And you can build professional quality drug discovery ecosystems. This is just one example built by Josh Horton, Inka Sheen, and Hugo McDermott-Peskin as part of the Antiviral Drug Discovery Center that I happen to lead, one of these 9 AVID centers working on pandemic preparedness. This is the entire workflow you can actually go and download and use yourself, for example, or adapt to your own drug discovery needs just by going to GitHub. Everything is open source. The cool thing is that industry is actually reporting successful use of these open source pipelines as well with reasonably good accuracy. And of course, we've been using this with great success in our new antiviral discovery programs as we build 2nd and 3rd generation inhibitors, for example, for SARS-CoV-2 and beyond. There's a huge consortium of industry that is behind this, both in terms of governance and technical steering, as well as sustainable funding. I think this is a great model for sustaining key features of the ecosystem. And of course, there's a huge number of folks that have contributed scientifically and otherwise as part of this. So with that, I'll remind you that my slides are available here if you want to look at any of the reading that I've linked to in these slides. And I'm very happy to take any questions. And I hope I've kept us on time.
You've kept ahead of time.
Excellent.
So thank you very much again for a very structured and highly rich presentation. You spoke fast, that's why you saved time. So a few questions that I'd like to read from the Q&A. You have alluded to the use of machine learning along with physics-based methods, but maybe given that you have time, expand on how you see this interplay between ML and physics-based methods going forward, especially that we're facing huge libraries.
That's a great question. There's several levels at which machine learning is going to be able to revolutionize this process. The first of which is that these campaigns are much more successful the more compounds you score. So one of the simple areas is that to avoid the cost of 12 GPU hours per transformation, can we score much larger sets of molecules by using machine learning surrogates in the loop, for example, to learn free energy-like accuracy for a new target without having to do every ligand transformation? So there's ample opportunity for using 2D and 3D ML methods for learning on each of these networks. This will save huge amounts of cloud computing time, perhaps tens of millions to hundreds of millions of dollars per year across the industry. So that's one opportunity. Another is to look at each one of these areas for which there's critical need to improve things. Obviously, folks are working to turn this force field parameterization problem into a machine learning problem where it can also learn from data as well as be trained on very large data sets. There's a movement towards building foundation models that use much more sophisticated functional forms for these force fields, like neural network potentials. Also, there's opportunities to improve on how we treat these chemical effects by doing a better integration of statistical mechanics with very clever, learnable machine learning approaches to Monte Carlo. And then finally, I think there's a huge amount of interest in areas like AlphaFold, OpenFold, folding flow, and all of these other variants that will predict multiple protein conformations and ideally multiple protein ligand conformations, integrating them into these simulations in a way that actually allows them to be Monte Carlo moves that sample from the right density is a huge open opportunity for which we could dramatically accelerate the accuracy and reliability of these calculations. So there's ample area in many places for machine learning to solve many of these problems.
So generally, you see a greater use of machine learning in the areas that historically have been only restricted.
Absolutely. A lot of these force field parameters came by human optimization rather than machine learning, which seems like a missed opportunity.
I think it's a quick question. The effect or importance of FEP calculations for the systems where a crystal structure of the bound pose is not available.
That's a great question. There is some work out there you'll see in recent papers trying to use things like AlphaFold to predict the protein structure and then docking to model in the ligand. Often there's some challenges with making sure the ligand will actually fit, and so sometimes you have to use induced fit docking. There's a lot of excitement about methods that co-predict the protein and ligand structure, and I think this is where a lot of the most enthusiasm is, because if you can do so with a reference ligand, then you're off to the races with these free energy calculations. So I think the answer is that we don't know yet, not exhaustively certainly, but there's a lot of optimism, especially from the gigadocking communities that have used very large scale, that's over 1 million compound docking to AlphaFold predicted structures that often there has been significant success there.
Thank you. So here is what I think is a loaded question that you may start answering and continue until tomorrow. The short version is, does it mean that no commercial tools are needed for drug discovery? My kind of extension to this question is, what is this, how do we play between industry-funded work and open work, who funds it, and how you started talking about it, how we make these tools validated, useful, and funded?
That's a great question.
And yes, question, are the gaps in open source tools available? That's an addendum.
Oh my goodness, yes, there's gaps in open source tooling. The hope here is that industry will have, first of all, there's a lot of utility for industry-produced software, right? Both in-house and through large commercial software vendors. But not everybody needs to invent everything from scratch, right? There's no need to have an entire ecosystem built from scratch. It's just very time-consuming for anyone who wants to get into this area, both academic and industrial. So the hope is to have a good, solid foundation, which we think of as these parts of the open source ecosystem, for which you can start with something that's a best practice that everybody can agree on, and then innovate in the areas where you want to have the best ideas or have the best contributions, or really want to focus your energy. And the way these tools are built is that everything is very modular. For example, you can take the open free energy tooling and replace the protocol object with your favorite free energy method, which may be proprietary, and it will drop right into the ecosystem that you can test along with other best practices methods. So I think there's a lot of opportunity for synergy and even for saving industry a lot of time in areas where it doesn't need to engineer everything. The current model that we've focused on right now is where we have industry-funded consortia, also with some academic led federal funding or nonprofit funding supporting these projects jointly. And I think that kind of model has been really successful for approaches like the Open Force Field Initiative, which have grown robustly under that kind of funding and a scientific input and governance model. Having both academics and industry folks help focus what they're delivering has been tremendously useful in making sure they're actually making tools that people can use.
Yeah, and my kind of hidden question was, should industry fund open science projects? And I think you just answered it.
Everybody should fund open science projects, Alex.
Assertively, but it also brings back to the industry useful tools.
It does.
So I believe that that's what kind of your answer is.
Yes, I agree.
What lessons were learned from the Moonshot project? What would you do differently?
That's a great question. We have done a few things differently as part of the Antiviral Drug Discovery Center, ASAP, that I lead. You can go to ASAP Discovery and learn about some of those things, that choices that we made that are different. One of them is that we had a lot of fun getting the entire community involved in crowdfunding early on, but it's certainly a lot of effort for everybody. And now you have actual jobs to go back to. We've tried to automate as much of this hit to lead as we can with AI and ML driven methods. Chodera has driven a lot of that hit to lead. So you can check out their recent papers on that from Alpha Lee, for example. The other part is we went without a patent in this case. And I think there is opportunity for having enough governmental funding focused on antiviral discovery in other cases where there's market failures, like Stephen Burley had indicated, in order for that model to be successful. But as we've seen recently, government funding for any kind of pandemic preparedness globally has really dried up. The NIH, as you'll recall, ended our antiviral drug discovery program 2 years earlier than expected. And so given that, it's very difficult to pursue things further. So what we've done instead to try to ensure we can achieve global, equitable, and affordable access is to have a new IP model where we will make sure that the patent will contain a single molecule and be licensed essentially for no cost as long as manufacturers agree to waive their manufacturing exclusivity rights, for example. You can read all about this on our website. But there are basically minor tweaks from all of our learnings that we've written more extensively about.
Thank you. So I'll translate the last question into a suggestion for you to aggregate, if you haven't done this yet, many delta G values computed with open source tools into a database, which I think may make sense to save people time on redoing something that's been done by others. But with that, I'd like to end the session. Thank you, John, for your excellent talk and all.
Thank you.
Other speakers. I think it was a fantastic session. We finished on time.